Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speaker, Chris Berger, Executive Director, Quality Viral Vector from Avid Bioservices.
All right, thank you, Alex. So today we're going to talk about Build It Right, a quality perspective on the design and build of a viral vector CDMO facility. And so at Avid Bioservices, we've been traditionally a mammalian provider of bulk drug substance, so doing monoclonal antibodies, proteins, and enzymes. About 2 years ago, we decided to get into the viral vector space. And this is a story from a quality perspective on some of the design decisions and path we took to build our new facility. And so really, our philosophy was that if we're going to do something, we're going to do it right, and we're going to start quality in the very beginning of the process. And so really, quality begins when you decide to manufacture advanced therapies. And in this talk, we'll go through scoping, stakeholder management, requirements, program management, managing the build, qualifying the build, and then strategy after we have the build complete. And so really we start with our business goal. As Stephen Covey says, begin with the end in mind. And the reason you do this is a few reasons. One is a quality reason, but the other are just practical reasons. You want to minimize change orders. You want to minimize rework. You want to increase the chance for project success, which is on time and on budget. And so really you want to start with some really broad scoping questions. For us, these were all viral vector specific, looking at what vector classes we wanted to be in the market for, what type of cell lines, were they human cell lines, primate, a mix, were the cells suspension cells, adherent cells, and then the scale of our operations. Are we looking at very small scale, mid scale, all the way up to a larger 2,000, 3,000 liter scales? And then our endpoint, are we looking to just manufacture drug substance, take it all the way through to drug product? Will we manufacture cell banks? Will we manufacture virus banks? We needed to answer these questions up front to get a very good idea of where we're headed. And then the next thing we did is really look at the team. And I know this sounds very rudimentary to a lot of folks probably on the line that have worked in these larger projects, but we found it very important to define the core team. Folks that are from start to finish, dedicated to the project, will be on every call to help with decision making. And then the extended team, or team members who come in to complete specific tasks that ebb and flow with the project. And so really looking at your core team of someone from manufacturing, someone from quality, someone from engineering, someone from IT, and then bringing in folks from facilities, automation, MSAT, business development, validation as needed. Really to kind of make sure that we have a core key group of stakeholders that are maintaining continuity of what the vision is versus different folks coming in with different questions, different thoughts, different ideas, and then the project shifting over time. And so really the bulk of this presentation is on our process for defining user requirements. And then once we have those, we'll go into some aspects of practicality on how we implement it and where we're at today. But for defining user requirements, we broke these down into three main groupings. One were general user requirements that are required for any biologics facility. The second were user requirements that were specific to viral vector production. And the third were unique considerations that were specific to us as a CDMO. And so these slides all kind of have the same layout. First, we'll talk about general, and then we'll move into viral vector, and then we'll move into CDMO. And so we won't spend a lot of time on general requirements. These are really the basics for any biologics facility doing GMP production. And so their ability to clean, adequate lighting, temperature, humidity, and ventilation, protection against insects and other animals, some level of access control, and then proper room classifications and pressure cascades, whether you're using the European grade system or the ISO system. At least having some of that in place is the basis of biologics production in a GMP clean room. Now we'll get into a lot more detail as we go through viral vector and CDMO specific considerations. And so the first thing we start with is HVAC design. At the highest level, you have a few questions. One is, are you using recirculated air or 100% outside air? And then the other question is, how do you zone your HVAC handlers? Are you using multiple HVAC systems, each with a dedicated room? Are you having one HVAC control multiple rooms? Are you having redundancy? Are you 100% redundant? Are you guaranteeing 100% uptime? And so some of our design considerations revolved around this diagram, which is a map of our clean room space. And so each color indicates a different air handler. And so on our clean room, we have 15,000 square feet of clean room, and we have about 8 to 9 different air handlers, all servicing different areas. Anything that's dedicated as a non-viral space is using recirculated air. Anything that's dedicated as a viral space is using 100% outside air. All the air handlers are fully redundant, meaning if any single air handler fan were to fail, there would be another fan in place that would be spun up to keep room pressurization. And so this is how we decided to do it based on risk, and that we wanted everything that was viral positive to be completely segregated throughout the facility, which leads us to our next topic, is segregation strategy. And so really there's two segregation strategies that you can use. One is separation in place, meaning that you have either physical boundaries within the room, separate incubators, separate bioreactors, or separate rooms. The other is separation in time, meaning that you use a shared space, but you separate activities through time, and that you would only have one product in a space at one time, open operations happening or occurring within that same space at different times. And so we did a combination approach. And so for viral positive zones, we're doing both separation in place and separation in time, meaning only one product available or in use in each of the spaces highlighted in pink. For viral negative zones, they're more of a shared space, and they can have multiple products and separated in bioreactors or incubators. And then really looking at biosafety level, how do you define your biosafety level? For most of our products that we would be making for viral vector, we would be using HEK cells, which are BSL-2 level. And so we defined a BSL-2 envelope, and that's indicated in the pink. And really looking at the BSL-2 envelope is just another level of safety. Some firms have specific requirements for spill containment within a BSL-2 suite. And so you're looking at zone borders, physical containment of spills, berms around the doors to make sure that if you have any spills, they're maintained within that BSL-2 envelope versus going into central or less controlled areas. And so again, this was a design decision based on risk. We could have made the entire facility BSL-2, but we decided that we wanted to have some level of spill containment in case we had a leaky bioreactor bag, leaking tubing, that we would have some level of containment to capture that liquid within that space. And so what we have are soft berms on the entry and exits within the material airlocks in and personnel airlocks in and out to contain any liquid that would spill within the area. And then next, we want to talk about decontamination strategy. And this is in addition to routine cleaning using microbial kill agents. And so what we looked at was vapor systems. And so typically these are vaporized hydrogen peroxide. We looked at that system, which uses a 40 to 50% hydrogen peroxide. And we also looked at technology that is produced by Tomy. It's called SteraMist. It uses an 8% mist versus the 40 to 50%. And so we do have the SteraMist system, and it's installed in all the viral positive suites. It'll be used in addition to routine daily, monthly, and quarterly cleaning with microbial agents and sporicide, and it'll be used really as part of changeover. And so whenever we finish a viral run in any of the suites, we would use this BHP IHP system to decontaminate the room as part of our product changeover before we bring in another client's project. And then lastly, we wanted to look at facility flows, personnel flows, material flows, and waste flows. And I will say that one of the lessons learned is you cannot spend enough time on these questions and spending time looking at how folks, materials, and product will move through the facility. We spent a lot of time here with different mock-ups of designs and thinking about how will we bring materials in? How will folks enter the building? How will folks leave the suites? How will materials leave the suites? How will product go from suite A to suite B to suite C? Really, spending the time here makes a facility more livable in the end. It's very easy to look at, a spaghetti chart and lines on a diagram and think, yeah, that makes sense. We can live in that until you see it built, and then you realize, well, we have a lot of excess flow here. We have a lot of paths that aren't that well thought out. And so the bulk of our design work, once we had an idea of where we were going from those scoping questions, was really on layout. How will folks move within the facility? How will raw materials enter the facility? How will product move throughout the suites and the production train? And so I think the take home here isn't so much what you see on the screen, it's that really spend time thinking about how folks are going to live in your facility. And then now we have CDMO specific considerations, and those are ones that we kind of spend some time thinking of. If you're an originator, you have different requirements at the end than if you're having folks coming in, touring your facility, and having person and plant. The reason I chose this picture, not because we're doing operations or anything medical, it reminds me of a Seinfeld episode where Jerry and Kramer are essentially person in a plant, and they're overwatching an operation on an observation deck, very similar. And then Kramer pulls out junior mints and he starts eating them, and then pretty soon him and Jerry get into a little arm fight, and one of the junior mints goes flying out and lands into the person. And so, not to say we would allow food in our clean rooms or anything like that, but the point is that Jerry and Kramer are essentially people in plant, and they're the worst type of people in plant, right? They're very disruptive, they're causing a lot of ruckus, they're breaking the rules. And so really having folks in your clean rooms is something that you have to consider. Are they allowed in the suites? Are they allowed just in viewing corridors? What level of training is required? And how will the team on the floor handle any interruptions or disruptions from having additional folks on site? And so one of the things that we did was we put in these pan-tilt zoom cameras in every suite. And these allow for folks to essentially be people in plant, but be outside of the clean room space. They have full control over the camera. It has a 360 view. It's movable by a joystick. They can zoom in. It has great resolution to the point where you can read, a batch record. You can read labels on raw materials if you zoom in enough. And really, they're throughout the suites to allow for folks on site to have an idea of what's happening in the suites. For critical operations, drug substance fill, a drug product fill, inoculation, we would allow folks in the clean room for real person and plant. But for the majority of operations, we feel that having local access with these cameras is enough to satisfy what most folks would get from person and plant. And then as a CDMO, the other thing that was very important to us was standardization of equipment. Really looking at where the industry is headed, getting equipment that is not quite bleeding edge, but really on the forefront of technology, things that can meet all data integrity requirements, all Alcoa requirements, have full audit trail from the electronic system perspective, but are also industry standard, that They're equipment that you would expect to see. They're equipment that you're probably using at your own sites. So that tech transfer is easy. We're not bringing onto equipment where we're worried about vendors going out of business or have to worry about consumable supplies or life cycle of these pieces of equipment that we chose. And it's always a challenge in choosing equipment for a CDMO in that you want to be industry standard. There's a lot of choices out there. And so really trying to be as agnostic as possible to what clients are bringing to us and having equipment and technology in place that could suit them. Of course, it doesn't work 100% of the time, and in those cases, we have to have a flexible plant where we can bring equipment in and out as needed. The next consideration was really multi-product manufacturing. How would we handle multiple products in the suite? Again, going back to the principles of separation in place and separation in time, we designed the facility to have two upstream trains, a train A and a train B, feeding the central downstream. And so within the upstream, if it's viral negative, we could have multiple clients of the same client, multiple products running. And then once it goes into the viral positive areas, it'll be a one-to-one, one product in the room, one client. And then having robust changeover and clearance procedures as we move clients and products through the facility. And so really it's a culmination of a lot of our design decisions of how the rooms are laid out, really looking at risk, looking at cross-contamination, doing layers of protection analysis, really to come up with robust procedures and SOPs to allow for multi-product manufacturing. And then looking at your regulatory alignment, are we looking at US only? Are we looking at the European Union? Are we just bound by US GMPs? Are we looking at UGLEX? Are we looking at support for the rest of the world? And so really looking at those requirements and just knowing upfront where we're headed really helps the engineering team design the proper pressure cascades, the proper airflow, making sure we have the correct sampling points for EM. All comes down to kind of, are we looking at Annex 1? Are we looking at just being, drug substance manufacturer? For our facility, we will produce drug product, and so we're fully aligned with all the requirements from the recently revised Annex 1. And then once we have an idea of where we are standing with scope, stakeholders, and requirements, we start to get into programming the space, managing the construction effort, qualifying the finished product, and then really looking at a strategy for maintaining compliance through the life cycle of our building. And so for programming the space, we're really designing according to our URS and our regulations. And so keeping in mind all those things we talked about, pressure testing each layout according to our requirements, looking at layers of protection analysis, looking at hazard analysis at each layout to say, is this something we can live with once it's built? Very easy to change design on paper, a lot harder to change design once it's built, even harder to change design once it's qualified and validated as a manufacturing facility. And the other thing that is really comes down to daily management is managing the contractors and the team doing the build on the floor. And so really making sure those conversations are well documented and that they're crystal clear, because a lot of times a hallway conversation can turn into a change order, which can turn into, best case is something you want, worst case is something you were thinking about, and then you show up a week later and you have a new door or a new thing in the facility that you weren't expecting just due to a hallway conversation. So really making sure you're talking clearly to those folks and they understand where we're headed from the build. Make sure you have a robust change control process in place to avoid any of those kind of changes that you weren't expecting. And then really managing the space, looking at comprehensive risk assessments, using tools, using a LOPA tool, using a hazard analysis, implementing risk mitigation strategies, and regularly monitoring and evaluating your risks. And then for qualification, this is standard validation team would come in. We start looking at installation, qualification, operational qualification, performance qualification, and then start validating the HVAC, the water systems, the BMS, EMS, system control software, and manufacturing equipment. This is really something that you start as building qualification, and then you continue on through the life of the facility with continuous requalification efforts. And then last, we'll go through this sort of quickly, but it's a controlled contamination strategy. It's introduced in Annex 1, which is effective in 2022, and it's used for the manufacture of sterile products. It's also best practice for low bioburden bulk drug substance. And really, the CCS is looking at external contamination, not product CQAs. And so what we look at is facilities and utilities, equipment, personnel, process flows, raw materials, container closures, cleaning, disinfection, monitoring, and then continuous management of these systems to ensure that you have a control strategy that will eliminate or reduce particulates and microbial ingress into your facility and into your products. And so the expectation is that this is a continuous program, not just separate SOPs that are kind of operating in tandem, but all put together and looked at on a quarterly basis, a yearly basis to say, how are we doing from a control perspective? How are we managing contamination? How well is our program working in whole? And so really this is hazard identification, control of the hazard, monitoring the hazard, and then verification that you have procedures in place to address those. And then on a continuous basis, looking at your EM, looking at your data, addressing gaps with CAPAs and corrective actions to ensure that you maintain on top of your system and that you're continually having good control of contamination events within your facility. And then when it's all said and done, hopefully you have a nice facility. This is a picture of our Abbott viral vector facility. It supports DS and drug product vector manufacturing. It's in compliance with US and EU regulations. It's cell line agnostic, it's vector agnostic. It allows multiple product trains for multi-product, fully segregated viral suites, BSL2. And so really we took a look at all our design efforts, and I think at the end we have a facility that we started with quality and we ended with quality, making sure that it was incorporated every step of the way versus a bolt-on at the end of the build. Thank you for your time.
Okay, great. Thank you, Chris. And as a reminder to our audience, you can go ahead and type in your questions for Chris now. So our first question is, typically construction projects have a finite budget. What tools did you utilize to assist in ranking design features and capabilities to remain with your allocated budget?
Yeah, so our priority was really safety, right? Safety for both our folks working in our plant and safety of the products that we were producing. And so we were able to just use standard risk assessment tools and really look at safety as our priority. We wanted everyone to be safe. We wanted to provide a safe working place, and we wanted to provide safe products to the marketplace. And so in some cases, there were things that we wanted to do from a footprint perspective or from an equipment perspective where we didn't have budget because we wanted to ensure safety was what we maximized in the build.
All right, and our next question is, in making decisions, did you rank viral vector considerations above CDMO considerations, or were they equivalent?
So viral vector considerations were ranked higher. Most of those came back down to safety and quality concerns. The CDMO considerations were nice to have after we met the viral vector considerations. And so things like the pan-tilt zoom cameras sort of fall into a safety aspect. And so those were prioritized, but by and large, the viral vector considerations were what we prioritized from a quality perspective.
And how do you manage waste materials from the production suites? Do you have to autoclave out materials?
So waste materials are decontaminated in the suite. Everything is disposable in this facility, so there is no need for an autoclave. We have whole time studies for how long things need to decontaminate, and so everything would be treated with bleach, and then solid waste would be handled by a waste handler. Liquid waste would go into the waste neutralization system before being led out to the municipal waste system. And so as
All
a
right.
part of the
Oh, sorry.
flow
Go
is
ahead.
we have exit hallway, everything, all the suites are unidirectional. And so materials come in through an airlock in, they leave through a material airlock out after they're decontaminated, and then they go through the exit hallway to leave the facility.
And let me just see here. How do you, did you validate decontamination of shared zone for viruses, especially very high resistant viruses such as AAVs?
Yeah, so we didn't per se go with the route of using model viruses. What we did was for our in-room fumigation system, we used geobacillus indicators to validate the cycle for the room. And then along with routine EM, we feel fairly confident that we would be killing all viruses in the room once all those geobacillus indicators have turned. And so that system is validated, and then we would continue to do periodic revalidation of that system as part of room changeover.
And do you record and save the video from the production cameras, and do you review video as part of batch disposition? If not, if you do not, have you ever had to justify to regulatory investigators of not reviewing these video records?
So our plan is that these videos would not be saved. They would be live feed to work just similar as a person implant, and so you would see what you see at the time that it's happening. We're not planning to record them. They're not for the purpose of determining quality events or part of batch disposition. They would just be kind of similar to what person implant would see. They're not security cameras. It's a different system for security within the facility. This is just trying to get one less person out of the clean room. And for the second part of the question, no, not yet, and hopefully not.
All right. Well, it looks like we're running just a bit short on time, so if we don't get to your question, we'll be passing them along to Chris, who can follow up with you directly. Please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, being a CDMO, how do you plan for multiple different types of equipment or process flows based on client demands?
Yeah, so really, I think for us, it's industry standard equipment and building a facility where you can bring equipment in and out of the facility without a complete shutdown. And so really that for us, it's material in airlocks, having large double doors that are tall enough to handle most equipment, and that we can bring in equipment as part of line clearance or product changeover, and then remove equipment from the suite just as easily. And so really it comes back down to design. Try to keep it as process and equipment agnostic as possible so that when we do see what clients are bringing us, we can adapt as needed.
All right. Well, thank you so much, Chris.
Thank you.
And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Bye.